Okay, so this is another tips and tricks video, and this video is going to cover 3 topics, but they're all thematically related. And so I guess I would consider this a tips and tricks on how to maintain your records, your information, and also your samples so that they can be useful to you as you do your work, and also useful to other people even after you're gone out of the lab. And so we're going to cover 3 things in this video. One is how to keep a lab notebook. The second is how to maintain your digital information, so on our server. And the last is how to store your physical reagents, like tubes and things, so that you can find things easily and other people can find things if they need them. So the first thing we're going to cover is lab notebooks. And I've made a little tutorial that I just totally made stuff up, this dingle moo assay. It's not real. But I can give you one of these copies if you'd like to see what I think are good examples of lab notebooks. So I can kind of show you this, but there's lots of detail. It references what protocol is being used. It's dated all the time. You can see that this is really well explained. There's even notes to yourself, so you should check this, or I didn't get this out until I was ready to use it, or where things are. It's okay if you make mistakes in your lab notebook. You usually want to just cross them out. And you also are going to note deviations from protocol. So a protocol might say incubate for 1 hour. And you talk to me or someone in the lab and you figure out that you can incubate for 1 1/2 hours because you have to go to class or something. So that kind of deviation needs to be noted in your notebook, so that if something weird happens, like it works better or doesn't work as well, you can go back and trace your steps. There's also a lot of information about samples. So if you have 4 samples, you note what those samples are, especially if they're color-coded in the rest of your notes. You might make drawings of how you set up protocols, especially when you're new to something. I remember when I was just starting out, I would draw a lot of things like how to set up a Western blot or things like that. You can write each step kind of numbered. You're going to include all of your results, which is one thing that a lot of people forget. So after you finish your experiment, what is the result of that experiment? It might be measurements or pictures of animals or pictures of gels in your records. And if there's digital information associated with this experiment, you'd want to list all of those file names. So say you were doing an experiment where you were going to do imaging. You would image your animals, you'd have files for each one of those images, and the names of those files would be in your notebook as well. So that's kind of a really excellent lab notebook. Really, you want, especially when you're starting out, and this would be especially true for rotation students or undergrads, more is better. You want really detailed notes, because this is not only a record of what you've done, but it's also a reference for yourself. So if you run a PCR, the second time you run a PCR, we might expect you to do it by yourself. And so if you've kept really great notes of where everything is and how to do everything and what everything looks like and how to label things, then you'll find that repeating that experiment will be very easy for you. If you've just, jotted down stuff on paper, you won't be able to repeat it. The other reason that lab notebooks are really critically important is for ethical reasons. So ideally, when you do experiments, you are going to come up with data and analysis and results that are going to be useful for us, and that we may even write papers about at some point in your tenure in the lab. And so we are not able to publish any results for which we do not have excellent records. So if you gave me a file and you said, this is an image of when I did a staining, an in situ hybridization staining, and this is what gene X's expression pattern looks like. If I don't have the records of when you did that, how you made that riboprobe, every single bit of it, I can't ethically publish your results. That's terrible, right? Because you've done all the work. You should get the credit. But unless you have all the records, it won't even matter. We can't do anything with it. It's totally like just lighting money and time on fire. So we really need these records for ethical reasons, so that you can yourself go back to them, so that you know how to do things the next time, and also so that other people can go and look at them. So other people should be able to look at your lab notebook and recreate what you've done, where things are, what files are, and so on. I will say that this page, I had also said that in a really excellent lab notebook, not only do you have what you did, but also some analysis. So after you did it, what the purpose was, what your results suggest, what you might do next, troubleshooting, and so on. So let's give you some examples of lab notebooks that are not quite as good. So this one's one that's like a little bit less detailed. Again, like I said, if you've done something 10 or 15 times, you may be able to kind of take some shortcuts. And if it's really close to a protocol we have, you might be able to back up to something like that. Again, please don't do this in the beginning, because you're not going to have enough information for yourself. And then people start to get a little bit lazy and take too many shortcuts and just jot things down. Leave out things. Where are these samples? Where did they come from? Where did you get them? How long did things last? What reagents did you use? All of that might be missing in someone who's just writing things down. Or sometimes people just say, see protocol. I did it like the protocol. Well, of course you didn't do it exactly like the protocol. Lots of our protocols say from 1 to 2 hours. So you need to write this down so that we know exactly what you did. And in addition to just writing lots of great notes, which you're going to do, another thing that's really important when you're keeping a lab notebook is to keep it organized. So if you just filed all of this away with no organization, you'd have all these great records, but it would be really hard to find things. You would have trouble finding things, and then I or someone else would definitely have trouble finding them. So we keep our lab notebooks in binders that look like this. We have tabbed pages. So like I like to have something that says, sorry, kind of awkward, planarian. So that might be my logs of my planarian feedings. I might have, what else do I have? Stocks. Every time I make a stock, I put it in that folder. If I'm doing cloning, I put it in that tab, and so on. And then if you've been in the lab long enough, you'll also have multiple volumes of these, so each one would have a number. I might have an index on the edge that says what all is in that lab notebook, the dates, and so on. And I ask that people, before they leave, also create a front page for each lab notebook that's a table of contents. And you can do this as you go, especially undergraduates and rotation students. I recommend that. Makes it easier to find things. So that's keeping a lab notebook. Again, just to emphasize, you want to write everything down. It's okay to have a little notebook to write things down while you're doing it, and then to copy all of that into a lab notebook. It's a little bit hard to write everything down as you're working, or sometimes I'll write things down prior to doing an experiment and then fill in the blanks of how long things take. Either one of those is okay, as long as in the end you have a record of exactly what you did in enough information that someone could easily repeat what you did. Okay, so that's part one, which is keeping a lab notebook. Again, I have an example that I can provide for you of just how I would keep records, especially in the beginning, and I'm happy to share that with you. The second part of keeping really excellent records is to keep a digital space with all of your files and digital information and writing projects and everything organized. So we have a shared research server that you should be able to get on when you join the lab, and you can talk to me or one of the senior members of the lab if you need help doing that. And on that server, you will have your own folder. And within that folder, you need to keep everything digital that you ever do in this lab. That means you need to have all of your lab meetings, like PowerPoint presentations or keynote presentations. You should have all of your posters that you make. So if you make a poster to present at a conference, that should be on there. You should have every piece of digital data that you ever create, which will mostly in this lab be sequencing files for when you're cloning, and imaging files. And all of those files need to be carefully named so that I can tell exactly what they are. So what I would say is all of those files should have the date, what that file is. So if it's a sequencing file, it may come with just like a random alphanumeric sequence or just your initials and A. You need to add to that what gene this is, what clone this is, what date this is, and so on. For your imaging files, it should say, what is this? So what gene did you image? What is the date when you did this experiment? What is the date when you imaged this sample? And so on. You might find that this is a little tedious at first because you're like, well, I could just find this stuff later. But as you're in the lab longer and longer, it becomes harder and harder to find things. So it's much easier if you put all of that information in the file name. That way, if you're looking through your notes and you realize, oh, I already did the in situ for this, I'm going to go look for that file, and you know the date, then you can find it. If you know the gene name, you can find it. If I'm looking through your records, I know what everything is and how to cross-check them with your laboratory notebook. So I would say all of your files should be organized. All of your data files should be organized. So you might have your folder on the server. It might have presentations, and all of your presentations would be in that folder. Posters, all of your posters would be in that folder. Sequencing information, and that might have multiple subfolders by date or things like that. Images, and you may have live image movies and in situs and things like that. The more organization you can build into your digital record keeping, it's just going to make your life a lot easier, and mine too. And I would say it's really important to start that process really early. You might think that, oh, well, I'll figure this out later, but the more data you have, the harder it is to get organized. So this is something that's really important to do from the beginning. I think that's about it for digital record keeping. One other thing I'll mention is that one of the actual physical products that most people generate when they're in the lab is a series of plasmids. So if you're asked to clone 10 genes, the product of that cloning would be 10 mini preps of plasmids that you've sequenced and that you know this is this gene, this is this gene, this is this gene. Usually we put these in PJC 53.2. What I ask for people to do, and I can give you an example of this or other people in the lab have it, is to generate an Excel spreadsheet that has all the information about those clones. So you would say, clone number 1, it's this gene, whatever gene you've cloned. And that might be a gene name. Usually we will have gene identifiers, which often look like DD underscore SMED underscore, so on. You may see these in the lab. Maybe that would be the third column. So first column is just a number. Second column is the gene name. The third column is the gene identifier. The fourth column is PJC 53.2, which would be the same for most of them. Then you might have the date, and then you would have the primers if you have that information, and then you would have the sequencing information as well, or the file where that sequencing is. So I should be able to, when you leave the lab, know every plasmid that you cloned, all this information about that plasmid. This is also much easier to do from the very beginning as soon as you join the lab than it is to try to keep all of this organized later on. So if you do this with your digital data, it makes it much easier for me to find things, for you to find things, and for us to make sure that there's continuity with the information that we have. And the reason that we keep this all on a server is that's backed up. A lot of people keep some of this on their own personal computer. If your computer's lost or stolen or stops working, then your data is lost, and that would be really devastating. So all of your data needs to be on a server that's copied and protected and things like that. And so that's another thing you want to start getting in the habit of early, is backing up your data. Okay, so we've covered laboratory notebooks. So that's your record keeping of what you do in the lab. We've covered digital record keeping, which is how you keep track of the files you generate and keep a digital record of all of the reagents you generate, which might be plasmids is the main one that comes to mind. But the other thing that needs to be organized during your time in the lab is your samples. So in the lab, you're going to do a lot of different things. You may do cloning, you may generate riboprobes, you may generate double-stranded RNA, you may generate PCR products, you may purify RNA, You may do any number of things, even things that we haven't even done yet, you may be responsible for doing. And so in the course of all of these things, you're going to generate physical products. And often in the molecular biology parts of the lab, you're going to have lots of tubes, just tubes on tubes on tubes. And we have some of these freezer boxes that look like this, that have racks. And one of the things you can do to keep organized is to make sure your boxes are labeled with your name. And then as you're in the lab longer, you might have your name, PCR products, your name, riboprobes, and so on, so that you can keep track of all of the different types of reagents you're generating. For the mini preps, and especially as you're generating one of these plasmid collections, as you sequence plasmids, you know they're correct and you want to keep them. You know, I have a set of plasmid collection boxes that are separate from my other boxes, and I just, as I put those into my Excel spreadsheet, I put them into a physical space as well. Okay, so that, if you keep these boxes organized, that will help you, but also you need to make sure you put information on tubes. So I have an example of, this is like a PCR product. I've just labeled a tube how I would label it. It says PCR, let's see, PCR1, gene X, and then the date on the lid. And then on the side I have my initials, and then I'm pretending I've taken the concentration of this with the spectrophotometer, and I would write that concentration on the tube too. So if I have this tube, I know exactly what it is. I know when I did it, so I could look at my lab notebook for more information. I know what it is. I know that it's a PCR product, which gene it is, and so on. What a lot of students and trainees do is they get in a hurry and they end up with a lot of tubes that look like this. Just one. And I don't know when this was created or what was created. I don't know if this is a PCR product. I don't know if it's a riboprobe. I don't know if it's the most important reagent the student has ever generated during their time in the lab. So this is a problem. Because this, to me, is just trash, right? If I found this, I have no idea what this is. And if you have a lot of tubes that are like this, you're going to have multiple number ones, right? Every time you do something, you'll have number one. So you might have a riboprobe and a PCR product and a double-stranded RNA that all look like this. And that's going to mean that you're going to be more likely to make errors in what you grab as well. As you generate things that you want to keep for a really long time, you may also use stickers. So in the first example, I've just written on the lid of the tube, but this is a little white sticker. So in the plasmid collection I mentioned, where I put things into a document and I know I've cloned this gene and I have sequenced it and I want to keep it forever, I also kind of create a little bit more of a permanent label. These markers, you know, Sharpies are great, but they do kind of fade over time as you open tubes. And if you get ethanol or stuff on stuff, it'll come right off. So these labels are a little bit more permanent. So in the end, what you should have is a collection of samples that are all really well labeled. Some of these things you may not have to keep for a very long time. So the mini preps that you keep in sequence, you want to keep basically forever, the whole time you're in the lab and beyond. If you create double-stranded RNA and then you've done your experiment and you don't need to use it again, you can throw that away when you're done. Riboprobes, after you know that you're not going to use them anymore, you can throw them away. PCR products, if you're not going to use them again, you can throw them away. But the mini preps are the things that you really, once you sequence them and know they're right, we want to keep forever. So you can also go back through your boxes periodically and say, okay, well, I have my PCR, maybe I have a box of PCR products. I'm going to go through and look and see if there's anything in there that I don't need anymore, and you can get rid of it. And so this, usually you'll keep your samples, all of these things we're talking about, in the freezer. You should have some dedicated freezer space, and you can talk to us about that. And we don't have enough space for everyone to keep everything indefinitely, though, so going through and keeping track of things and culling things periodically is really important. Okay, so those things all together, if you're able to accomplish that, which you should be able to, you will have physical records in your lab notebook of all of your activities in the lab. Those need to stay in the lab. They can't go home with you. You will have digital records on the server and backed up of all of your data and all of your records and all of your presentations and figures. And eventually when you're writing papers, those should go there too. And you should have a physical space that's organized into boxes that are well labeled, and your samples should be well labeled. And with all of that together, especially if you start that out in the beginning, you'll be in really great shape to make fewer mistakes, to have the records and things you need so that we can publish your work eventually, and also so that you can behave ethically, because all of this record keeping is not only just a practical issue in the lab, but also a really important ethical issue. We can't reproduce experiments or validate your results if we can't find things or know exactly how you did your work. So all that stuff's pretty important. I know you'll be capable of it. Please ask questions about this stuff soon after joining the lab if you have any questions. This stuff, while it seems kind of like less exciting or essential than some of the experimental stuff, this is really foundational information that you really need to get right, both in our lab and in other labs as well. Okay, thanks for listening.